Welcome to the Pancreatic Cancer Action Network's webinar, Immunotherapy for Pancreatic Cancer. My name is Sheila Bott, and I'm the Manager of Scientific and Medical Affairs for the organization. At the Pancreatic Cancer Action Network, our goal is to give you hope through better resources so you can make informed decisions. Patient Central can provide free, personalized information on many topics, including pancreatic cancer specialists and treatment options. Contact Patient Central at 877-2-PanCAN or email patientcentral@pancan.org. I would also like to take this opportunity to thank our webinar sponsor, Ipsen, and to thank our scientific and medical affairs industry members, AbbVie, AngioDynamics, AstraZeneca, Ipsen, Janssen Oncology, Pfizer, Rafael Pharmaceuticals, TriSalus, and TYME. The webinar is scheduled for 60 minutes and includes a presentation followed by questions, time permitting. The questions that are relevant to today's topic and more general in nature will be prioritized. If you have very specific questions, it would be best to address them directly with your healthcare provider. A recording of this presentation and the slides will be available under the Educational Events page at pancan.org. Now I would like to introduce our presenter. Dr. Gregory Beatty is an assistant professor and director of translational research of the Pancreatic Cancer Research Center at the University of Pennsylvania in Philadelphia. The mission of Dr. Beatty's research is to define mechanisms of resistance and response to cancer immunotherapy for informing effective strategies to condition tumors and patients for enhanced therapeutic responsiveness. He is a member of the Scientific and Medical Advisory Board for PanCAN, and he is also the principal investigator of the clinical trial consortium site for PanCAN's Precision Promise at the Perelman School of Medicine at the University of Pennsylvania. Dr. Beatty, I will turn the presentation over to you.
Thank you very much. It's great to have the opportunity to speak with everyone today through this webinar. Today I'd like to discuss immunotherapy for pancreatic cancer. I've been working on pancreatic cancer now for the past 13 years, and there are two major types of pancreatic cancer. The main type is adenocarcinoma, which represents 90% of all pancreatic cancers. The second major type is neuroendocrine tumors, representing about 10%. Today, I will discuss our understanding of the role of the immune system in pancreatic adenocarcinoma. So the immune system is a remarkable feature of our body. The immune system protects us from foreign pathogens, viruses, and bacteria. The immune system is also involved in wound healing. It can help in the initial point of wound healing by fighting infection, but it's also important in repairing tissue. So it's very important remodeling the tissue to make it strong again. The immune system is also important for learning and memory. The immune system is in our brain. The immune system actually patrols all of our tissues in the body and is constantly at work. But what is its role in cancer? The concept that the immune system could fight against cancer has been around for more than 50 years. And this idea has been formulated as what we call the Immunosurveillance hypothesis in cancer. And what it states is that a major function of the immune system is to control the outgrowth of cancer cells and normal cells in the body. And herein lies the premise of self versus non-self. In other words, that a normal cell that turns into a cancer cell is no longer identified as self. It is foreign to the immune system. The immune system has not seen it before. And overall, this hypothesis is now changing the way in which we treat patients with cancer. Immunotherapy, or the use of one's own immune system as a therapy, is transforming our way in which we treat cancer. It's a standard of care now for many cancers. Many of those are listed on this screen. Immunotherapy can produce durable outcomes with improved survival well beyond 5 years for some patients. Even some patients with pancreatic cancer can respond to immunotherapy, although most don't. The major challenges that we are facing now is trying to understand who will respond, and for those who do not respond, why. And to answer these questions, we really need to understand the nuts and bolts of how the immune system works. How does the immune system recognize and respond to cancer? The immune system is the ultimate form of personalized medicine. We talk about finding the right drugs for the right person. This is personalized medicine. But the immune system is also personalized. It is unique to each and every one of us. Our immune systems are educated by what we are exposed to. The infections that we get, the bacteria that live in our gut, the foods that we eat, the air that we breathe. Our immune system is continuously educated. But we are trying to convince the immune system to attack cancer. And the immune system has several features that make it a logical choice for cancer therapy. These are listed on this slide. For example, diversity and specificity. The immune system is capable of recognizing and causing immune responses against an unlimited array of targets, or what we call proteins. Proteins are present within cells. The way in which the immune system does this is it utilizes some specific cells, some of those called T cells, and others called B cells. B cells make antibodies. Antibodies are what we use to prevent recurrence of infections. We get a flu vaccine every year in order to generate antibodies that will prevent us from getting the flu. This, in essence, allows for the immune system to be a personalized medicine. Now, there are some unique features about the immune system, in that it can be a living drug, and that these cells that are called T cells and B cells can rapidly expand, and they can keep pace with a pathogen being a virus or a bacteria. So they can expand and grow in our body and to help fight infection, but could also be used to help fight cancer. One of the most remarkable features of the immune system is its ability to remember, memory. The immune system can rapidly respond to things that it's seen previously, these previous encounters. And that's the rationale behind vaccines. There are challenges, though, that exist with the immune system. And these are these last 2 points, appropriateness and tolerance. Not every response that the immune system makes is the same. It depends on the type of insult that the immune system has encountered. This allows the immune system to be quite pliable. That means that the immune response to an allergen or allergies is a little bit different than our immune response to a virus. Now, the immune system also has to be tolerant, because it can't just go around recognizing everything in our body. It has to be able to distinguish between self and non-self. If it were to recognize self, our normal tissues, that would lead to an autoimmune disease. And so there's a balance between how activated and how suppressed the immune response needs to be. This also brings out some of the key points of how immune therapy might affect a patient. And that's because the side effects that come with immunotherapy are different than those types of side effects that we would see with chemotherapy. And these side effects that we see with immune therapy reflect immune activation and the release of proteins that manifest often as a fever or chills or sometimes fatigue, tiredness. Immune activation can also lead to the immune system causing damage to normal organs, such as the gut, leading to colitis and diarrhea. It can attack the skin, leading to a rash. It can attack the liver, leading to a hepatitis. It could even attack the lung, causing shortness of breath and a pneumonitis. And there are other organs that can be attacked by the immune system. In many cases, though, these side effects can be managed, but they can also be deadly for a select few, if not controlled. So what does the immune system look like when it's in action? This video here will show you this video was to show you an example of what the immune system looks like when it tries to attack cancer. What you can see here is the immune system in green attacking cancer cells in the Petri dish in the laboratory. And when it attacks those cancer cells, they turn red. And what you can see from this video is that the one or two immune cells, in this case T cells, can very rapidly kill many cancer cells. They're almost like serial killers. Now we can manage to teach the immune system to kill cancer cells very easily in the laboratory. But how do we leverage the immune system to fight pancreatic cancer in people? This might seem like it should be straightforward, but we've encountered many obstacles that form the basis for ongoing research. Simply put, cancer growth is dependent on two features: how fast the cancer multiplies, or the growth, and how fast it dies, or death. And when it multiplies faster than it dies, that leads to accumulation, which means that the cancer is getting bigger. What we'd like to do is to slow the cancer growth rate, but we would also like to be able to increase the death rate. And together, if we can do that, then we should be able to cause the cancer to shrink. The immune system can be trained to do both of these jobs. It can be instructed to suppress cancer growth, but it can also be trained to kill cancer cells. And together, that should lead to tumor shrinkage. So what does this look like in more detail? Well, a major cell type that I've already referred to is in the immune system is called the T cell. And we and many others have been focusing on this particular cell type in the immune system as a major effector for recognizing and eliminating cancer cells. Now, how this T cell recognizes cancer is through a specific receptor. It's called the T cell receptor, or TCR. And that receptor is capable of recognizing proteins or distinct components of a cancer cell that are broken down and presented on the surface of the cancer cell using a molecule called an MHC. This creates an MHC peptide complex. This unique feature that is presented by the cancer cell can be recognized by the immune system. And when it does so, that can lead to cancer cell death. And what we'd like to be able to do is to make more cancer-killing T cells. And of course, we'd also like to be able to make those cancer-killing T cells work better. A couple of examples of how to do this are shown here. The first is that we could use vaccines to educate the immune system to make more T cells. We could also genetically engineer immune cells, even T cells, so-called CAR T cells that you may have heard of. Those CAR T cells, or the T cells, could be used to attack cancer. We can also remove the brakes from the immune system. That's tolerance that I was talking about, using drugs that will block immune checkpoints. But we can also try to step on the gas by using viruses to actually activate the immune system, or other drugs that can activate and get the T cells going. And there are many more strategies that are in development. So let's take a closer look at some of the details that shape how cancer is recognized by the immune system and that have formed the basis for current FDA-approved drugs. Now, there are several steps that are involved. The first step is that the cancer must have proteins that are unique to it. These are the non-self proteins, things that the immune system has never seen before. Those proteins have to be taken up by specialized cells within the immune system, so-called dendritic cells, which will present those to these T cells. In this part of the immune response, this is called the priming phase. It's getting things going. It's getting the T cells activated. And there are several signals that help to regulate this process that form the basis for FDA-approved drugs. There are signals that help make this process work better, but there are also signals that try to limit how effective this initial step in the immune system is. And one of those proteins is called CTLA4. And antibodies have now been developed against this protein. And by blocking this protein, T cells can expand much better against a protein, including a protein that's presented by cancer cells. Now, after those T cells are primed, they must expand and they must traffic. They have to get to the tumor. And when they get to the tumor, there's another phase, which is called the effector phase. This is when the T cells want to engage with the cancer cell and try to kill it. And the immune system has several checkpoints here too that try to limit the ability of the immune system to target tissues. This is to control and try to prevent autoimmune responses from occurring. And there have now been antibodies that have been developed that are FDA approved that try to block some of these negative signals that aim to dampen the immune response within tissues. And one of these is against PD-L1, and other antibodies have been developed against PD-1. And both of these are used now in patients with cancer. Overall, this science is what's driving new treatment strategies and has led to these clinical-grade antibodies that can block CTLA4, PD-1, and PD-L1. Now, cancer immunotherapy has shown remarkable success for some cancers. One example is shown here of melanoma, where nearly 50% of patients can respond to immunotherapy. But we have seen that this responsiveness is not the same for every cancer. In fact, most patients with pancreatic cancer do not respond. But there is a subset of patients, less than 1% of all pancreatic cancer patients, that do respond. Who are these patients? Well, some of them happen to be patients that can be identified genetically by looking at the genes in their cancer. And those genes will tell us whether or not that cancer has many, many more non-self proteins, mutations that make it look much different than a normal cell in the body. And it's those patients which have a cancer that's called microsatellite instability high tumors, or MSI high, that we have been able to identify that responds to some of these FDA-approved immune therapies. And one therapy is approved, it's pembrolizumab, for mismatch repair deficient tumors, or the MSI high tumors, including pancreatic cancer. But still, that means that 99% of patients are not responding. And so why is pancreatic cancer so hard to treat with immunotherapy? Well, there are many reasons to this. Some of those are listed on this slide. The first is that very often pancreatic cancer is detected late. It appears that pancreatic cancer has developed strategies to make itself invisible to the immune system. Sometimes this means that it just looks like normal cells in the body, even though it's not. It doesn't have those mutations, as many as we see in other cancers like melanoma and lung cancer. And sometimes it's because the pancreatic cancer cells have found ways in which to not present those unique differences that it has to the immune system. And in essence, it has a cloak over itself, allowing it to remain invisible. The other reason is that the immune system is not always a good thing for pancreatic cancer. In fact, inflammation or pancreatitis, inflammation in the pancreas, can drive the development of pancreatic cancer. And so having the right flavor of an immune response against pancreatic cancer is important. Else, it can actually act as an accelerator and help the cancer to grow. Now, when one looks at the microenvironment, the area that surrounds pancreatic cancer cells, this microenvironment can be very hostile as well. In many cases, it doesn't have the oxygen, the nutrients that are needed for the immune system to be able to thrive. It has a neighborhood that is not beneficial or conducive to the immune system. And we've also been learning that the host, or the patient who has pancreatic cancer, that sometimes the immune system may not be quite as fit. We don't know exactly why that's the case, but what we have learned is that when we take cells out of a patient with pancreatic cancer, specifically T cells, that they don't always divide as well as they would from a patient who was otherwise healthy. We're trying to understand why that might be. Now, this lack of success of immunotherapy, pancreatic cancer thus far, has caused the community to have to rethink this problem. How do we get the immune system to attack pancreatic cancer? Well, first place to start might be to ask what is different between those cancers that respond to immunotherapy and pancreatic cancer. And when one looks at the pancreatic cancer under the microscope, one can see some glaring features that make it look much different than to other cancers. Here are 3 photographs of cancer. The 2 on the left are pancreatic cancer, and the one on the right is lung cancer. And what should be immediately apparent is that the left photograph has few brown dots. The middle has brown dots, but they're focally distributed. And the right has brown dots diffusely distributed. Now, these brown dots are T cells. They're the major effector cells of the immune system. And the yellow areas are the cancer. What these photographs really show is that T cells sometimes never make their way into pancreatic cancer. The cancer remains invisible. In other situations, they end up only in some regions. And this is in stark contrast to what we see in lung cancer. And I think it's important to note that the T cell distribution that's seen here in this lung cancer photograph is exactly what is needed for immunotherapy to be effective. The T cells need to get into the tumor to engage the tumor, recognize the tumor, and then kill it. And we almost invariably don't see this in pancreatic cancer. In my own laboratory, we are thinking about what prevents the immune system from working against pancreatic cancer. This is a photograph of pancreatic cancer. It shows many of the immune cells that are present in cancer. And if we dig down into this tissue, we can start to see that these cells are coming in close contact with the cancer, which is in yellow. There are many types of cells within this tissue. This neighborhood has the cancer cell, which is in yellow. It has the T cells, which are in teal. And one thing to note is that many of these T cells are actually not touching the cancer cell. For whatever reason, they don't seem to come in direct contact. You also find macrophages. These are the big eaters of the immune system. They like to eat things. They can eat the matrix. They can help to remodel it. They can help cancer cells to move throughout this neighborhood. There's also regulatory T cells, which are designed to try to dampen the ability of T cells to work in tissues. And there are other cells, things called fibroblasts, to help to hold the neighborhood together, to create some strength to this bed of matrix that surrounds these tumors. Now, how we are thinking about the immune system is much like the game of Jenga. In Jenga, wooden blocks are arranged to form a tower. And this is analogous to forming a wall that would impair the ability of the immune system to attack what is on the other side. And what we need to do is to remove wooden blocks or elements of the microenvironment to create a neighborhood that is more hospitable and accessible to the immune system. If we can successfully identify the key the keystones that form this wall and remove them, we could crumble the immune barrier that pancreatic cancer has devised and allow the immunotherapy to work. Now, manipulating the immune system in pancreatic cancer is actually quite challenging. There are elements that may help the cancer, and there are elements of the immune system that may suppress the cancer. For example, the immune system can help pancreatic cancer spread throughout the body. And this biology can be seen in this photograph here. These individual regions that I've highlighted here we'll take a closer look at. So there are driver genes that help define the biology of pancreatic cancer. Some of those are listed here: KRAS, P16, P53, and SMAD4. KRAS is nearly present in 95% of all pancreatic cancers, and it has been found in the laboratory to be a key element of the ability of pancreatic cancer to grow. In fact, if we could eliminate KRAS or block its activity, it has been shown that we could cause pancreatic cancer to shrink. And animal models have shown that. However, this gene, this protein, KRAS, has been very difficult to target. There are studies now that are looking at drugs that can target KRAS, but not every KRAS is actually the same either. And these new drugs are not necessarily targeting all the types of KRAS that are drivers of pancreatic cancer. And we've also learned that there are resistance mechanisms that can emerge even when KRAS is targeted, such that it may not be the only solution to targeting pancreatic cancer. In fact, each of these common genetic aberrations that are shown here can be a contributor to the desire of pancreatic cancer to spread. What you can see from this figure here is that there's a cancer cell that's now trying to escape into the neighborhood. And there are many factors within this neighborhood that can cause this to happen. Some of these are these fibroblasts that hold the neighborhood together. Others include a low oxygen content. It's almost as if the cancer cells are trying to escape to go try to find nutrients that are needed for it to grow. And once the cancer cell does escape into the neighborhood, there are other cells that are present, in this case macrophages, that are shown here in brown, which can help to pave a path to allow these cancer cells to move through this matrix in search of a blood vessel to go to a new tissue or organ. As cancer cells move through this neighborhood, they also must evade the immune system. They must escape recognition by T cells, and macrophages can help to do this. You can see this brown macrophage almost sequestering the T cell and teal from interacting with the yellow cancer cell. In animal models, we've learned that when there are fewer T cells present, that this correlates with an increased risk for spread to other organs or metastasis. And tumors that lack T cells in patients also show a higher metastatic potential. Once the cancer cell is on its way through this neighborhood, it seeks to find a blood vessel to go to another organ. And when it does so, it encounters macrophages in the blood vessels, which help to cooperate to allow this cancer cell to get into the bloodstream. So the immune system in cancer, it's a balance. It's not just that the immune system is around to attack cancer. The immune system can also, in some cases, help cancer. So on the one side, the immune system is poised to attack cancer, yet cancer has devised strategies to escape elimination. And on the other side, the cancer has learned to leverage elements of the immune system to help it grow and to spread. My laboratory and others are actively studying this biology of how pancreatic cancer spreads. And what we've learned is that cancer can educate other organs to be receptive to these disseminated cancer cells. And this involves the immune system. In fact, inflammation alone is sufficient to drive this biology in animal models. And one key element is a protein called interleukin-6, or IL-6. Increases in this protein are associated with obesity, with diabetes, and both are risk factors for pancreatic cancer. It's also associated with cardiovascular disease. And what we found in the laboratory is that this interleukin-6 is a key factor that can support cancer spread to the liver, the most common organ site for pancreatic cancer to go to. Research is underway now to find ways to prevent cancer from educating the liver and other organs to be receptive, to prevent their receptiveness to cancer spread. We're also trying to find ways in which to prevent cancer from growing if it reaches a distant organ. If we can't make it go away, is there a way to make it stay dormant so that we could live with it, just like other managed illnesses such as diabetes? Ultimately, though, we would like to make pancreatic cancer go away for sure, and the immune system has the potential to do that. Designing strategies to convince the immune system to eliminate cancer requires an understanding of the steps that are needed to do this. We've reviewed some of these already today, what it takes to get the immune system to recognize cancer. This is a multi-step process. There have to be proteins that are non-self, unique, and have not been seen by the immune system. Those proteins have to be released by the cancer and taken up by specialized cells within the body that can present them to T cells. And that leads to this priming phase. The T cells get activated. And then they have to traffic. They have to go into the bloodstream and find the cancer. And once they get there, they have to infiltrate, go into the cancer. And once they get in the cancer, then they actually have to recognize the cancer and kill it. And as a community, we have identified drugs that we can use to make each of these steps work better. And many of those are listed on this slide. But what we haven't figured out yet how to do is how to put all of these pieces together. Answering this question requires reflecting on how pancreatic cancer is treated and how the immune system can be incorporated into this treatment strategy. So our current standard of care for pancreatic cancer relies on chemotherapy or cytotoxic therapy. The two regimens that are most commonly used are first drugs, a combination of drugs called gemcitabine and nab-paclitaxel, and a second combination of drugs that involves 4 drugs that we commonly call FOLFIRINOX. And typically our approach is to continue treatments until resistance develops or the patient receiving the treatment is intolerant of that treatment regimen. These treatment strategies can be quite effective in controlling pancreatic cancer for some time, but we do know that for the majority of patients, they ultimately will fail. And so we must do better. Many ongoing strategies are trying to combine immunotherapy with chemotherapy to help improve how long the treatments work for. And there is good rationale for why to do this. We have learned in the laboratory that chemotherapy can activate the immune system. It can do so by releasing those proteins that are non-self that can be recognized by the immune system. We've also learned that immunotherapy can make chemotherapy sometimes work much better. And so there's good rationale for wanting to combine these. And some examples of immunotherapy that is being used in this context include vaccines, as well as these immune FDA-approved drugs, PD-1, PD-L1, and CTLA-4, and other strategies designed to really step on the gas to get the immune system going, such as an antibody against CD40. But are there other options that we could consider for how to incorporate immunotherapy into pancreatic cancer? As a field, we are slowly moving to a new approach, where chemotherapy may serve as the initial strategy to gather control of the cancer, a so-called induction therapy. But then we use other strategies to maintain that control. In essence, our goal is to coax pancreatic cancer into remission, and it may take several therapies to do that. Timing matters. When do we deliver immunotherapy in this sequence? Do we wait until the cancer is no longer responding to the prior treatments, or should we incorporate it when the cancer is vulnerable? Well, one approach is to incorporate immunotherapy with chemotherapy, but an emerging strategy is actually to think about how can we switch to a maintenance immunotherapy. Once we've gathered control and presumably have activated the immune system, can we use the immune system now to control the cancer and maybe even put it into remission? We also have to think about what are the combinations of drugs that we want to use. Which ones do we choose? And for how long do we use them? Sequencing also matters. How do we coax the cancer into remission? What is the order of therapies that are needed? Can we use some treatments to shift pancreatic cancer from immune resistant to immune sensitive? Well, there are a number of strategies that are being considered for doing just this. These are emerging from the laboratory. And they are targets that are being pursued to tame pancreatic cancer and to leverage the immune system in the fight against cancer. Some key principles that are guiding this effort are shown here. For example, we'd like to have strategies that can sensitize cancer to immunotherapy. We'd also like to be able to remove elements of immune suppression. We'd like to be able to enhance the visibility of pancreatic cancer to the immune system. We'd like to be able to improve the cancer killing potential of the immune system. And it's possible that we may need to engineer the immune system to attack cancer. But there are so many targets and so many possibilities that are listed here. And how many targets do we need to engage, and in what sequence? The possibilities really are daunting. We need an approach to be able to tackle this problem. And so to do this, we need reproducible model systems that can help guide us on what we should be testing in the clinic. These model systems serve to inform cancer biology, treatment response and resistance, and also aim to help us design and develop new therapeutics. There are really three major approaches that are being used, and these involve taking cancer cells and putting them into animal models, using the cancer cells themselves, or studying the tissues, somewhat like I've already shown you in terms of the photographs, to try to get a snapshot of what's going on in the tissue and how the immune system and cancer are communicating with each other. Animal models are using cancer cells that come from patients. We've also developed animal models in which animals spontaneously develop pancreatic cancer. In many of these models, the cancer looks like under the microscope, just like human pancreatic cancer, such that the pathologist, one who looks at this all the time under the microscope, really can't tell the difference between an animal-derived pancreatic cancer and a human pancreatic cancer. These models serve as very valuable resources because they can include an active and functional immune system to allow us to be able to test drugs that would activate the immune system against cancer. Cancer cells can also be grown in the laboratory. And you may have heard of things called organoids. Organoids are three-dimensional constructs of pancreatic cancer, where these cells are now growing like little apples or cubes or spheres. We can also grow these cancer cells in the Petri dish and try to understand how, when we add various components of the immune system, How do those immune cells engage cancer cells? And how do cancer cells learn to resist elimination by the immune system? And finally, we've also begun to take tissues from pancreatic cancer biopsies and resected specimens, and we can slice those tissues now and try to understand what does that biology look like in the laboratory. Those tissues can stay alive for days, and that gives us an opportunity to study these interactions between the immune system and cancer in the laboratory. So there are many things that we can learn from this approach. Overall, these approaches are teaching us about the combinations and sequences of drugs that we can deliver. We've learned that drug A plus drug B then is not always quite the same as giving drug A, then drug B, or for that matter, giving drug B first and then drug A. This indicates that we need to understand the combinations that we're going to be delivering to patients, and these models provide that engine for leading to novel clinical trials. Now, this slide shows some recent data from clinical trials testing immunotherapy alone. So there's no chemotherapy involved. It is noteworthy here that the patients who received these therapies had already received prior chemotherapy. And so in some cases, they've even received several other therapies. As a result, these studies essentially are identifying a small subset of patients. Now, while few patients overall responded to these treatment combinations, it is really important to note that some patients actually do. And this indicates that immunotherapy can be effective for some patients with pancreatic cancer. But what we need to know is for whom. And again, for those who do not respond, why? Overall, this provides hope, though, that the immune system can be convinced to attack pancreatic cancer. Now, this uncertainty that exists around how a patient will respond to standard of care and to immunotherapy has led to a revolution that focuses on improving our understanding of the biology that predicts outcomes to therapy. Understanding the genetics of pancreatic cancer has clearly led the way, and we have identified as a community several genes that are actionable targets for pancreatic cancer. Some of those are listed here. But these genes comprise only a small group of pancreatic cancer patients, and still not all patients respond to these therapies that target these genes, and responses are not always durable. Recent interest has also flurried around the importance of the microbiome, the bacteria, the fungi, and other microbes that play key roles in our health. It is clear that many factors can shape the microbiome of a person, and that the microbiome that is seen in the skin, in the gut, in the mouth, and even in cancer can vary significantly and can have implications. The microbiome has become an exciting area of research in pancreatic cancer, and what we have learned is that certain flavors of the microbiome appear to correlate with distinct immune responses to cancer, and that this can have implications on the success of immunotherapy in other cancers. But what we don't know right now is how to manipulate the microbiome successfully for the treatment of pancreatic cancer. Now, the immune infiltrates in pancreatic cancer is also a key determinant of outcomes for patients. We've talked a little bit about this. T-cell infiltrates associates with improved prognosis for patients. So those patients who have resected cancers and T-cells are found in high numbers, they have a much higher likelihood of having a durable tumor-free outcome. But the same holds true that's a predominance of inflammation. So an absence of T cells, but those other inflammatory cells can drive a worse prognosis or outcome for patients. We are hopeful that these types of findings showing either more T cells or more inflammatory cells will help to give us a more detailed understanding of the immune system in pancreatic cancer and to help us identify those patients who may respond to immunotherapy. Overall, the goal here is to identify biomarkers that can inform the prognosis, the treatment selection, and the likelihood of response for patients. These biomarkers may come from biopsies, tissue biopsies, blood collection, stool, or even saliva. Overall, immunotherapy, though, has garnered significant interest among doctors and patients. And currently, there are over 50 clinical trials that focus on immunotherapy for pancreatic cancer, and more are being considered each week. This is a high priority, and it remains so because of the precedence that immunotherapy can produce durable responses, even leading to complete remissions of cancer. Well, we need to learn how to do this for pancreatic cancer, and clinical trials are how this will happen. However, less than 10% of patients with pancreatic cancer are typically enrolled on clinical trials, a number that needs to improve. And these trials are being conducted in patients before they go to surgery, so neoadjuvant, after surgery to prevent cancer from recurring, and in patients with metastatic or unresectable cancer. Many of these trials are leveraging drugs that are FDA approved for other cancers, namely those that block PD-L1, PD-1, or CTLA-4. And some are being combined with chemotherapy, as we've talked about, as well as radiation. And there are even new approaches to engineering one's own immune system, so-called cellular immunotherapy, that are being explored. This is a strategy that is working for some patients with lymphoma and leukemia, yet we have not yet seen reproducible and significant benefits in patients with pancreatic cancer. There are also new and improved vaccine strategies that are being developed and tested to leverage strategies that leverage viruses to attack cancer and alert the immune system. There are a number of targets as well that are being addressed, and some of those are listed here. Overall, there is a lot of excitement around the potential of immunotherapy in pancreatic cancer. And while we have a ways to go, I think we are on the right track. So the immune system is an attractive target for pancreatic cancer. It is the ultimate form of personalized medicine, and it is a living drug. The immune system, though, is based on a balance of signals that define its outcome. Whereas it can help cancer grow, it can also be educated to treat cancer. Some patients with pancreatic cancer do respond to immunotherapy, but most do not. We need to understand better why pancreatic cancer is so resistant to immunotherapy. And science is paving the way for new treatment strategies that will overcome resistance and harness the immune system for clinical benefits. Clinical trials are fundamental, making immunotherapy a viable option for patients. And while we are not there yet, there is strong hope that immunotherapy will change pancreatic cancer forever. Our goal is to make immunotherapy an option for all, not just a few. I want to thank you for joining this webinar and your interest in immunotherapy for pancreatic cancer.
Thank you, Dr. Beatty, for a very informative presentation. We do have time for questions, so I'd like to go ahead and start off with the first one. Does having molecular profiling help to determine if immunotherapy would be an effective form of treatment?
So molecular profiling can identify certain genes that do associate with improved outcomes to immunotherapy. One of those is the ability of molecular profiling to identify patients who have mismatch repair deficient, or what I called earlier, microsatellite instability high tumors. That's MSI high tumors. Those patients that do have that particular genetic feature to their cancer can respond to these FDA-approved immune therapy drugs. In fact, more than 50% of patients can respond, and those responses can be quite durable for patients. So yes, the answer is that molecular profiling can be used and is being used and should be tested on every patient to identify that small group of patients who do have that particular genetic feature, because it can offer a very important and effective immune therapy treatment option.
Great, thank you. The next question is, are there any supplements one can take to help boost their immune system or promote healthy immune cells?
So this is a very common question that many ask. And I think, the answer to this question is that we don't really know the precise types of supplements, the precise type of diets that are needed to make the immune system more effective against pancreatic cancer or even other cancers. An important component, though, is that the healthier you are, the better you do. And so what that means is that exercising, eating a healthy, well-balanced diet, getting good sleep are very key elements. Staying happy, involved in as many activities as you possibly can, leading a normal life as best you can in the setting of the cancer is very important. And those patients who do that tend to do the best overall. All of those things that I just shared, sleep, exercise, and eating a balanced meal, affect the immune system. They can make it much more effective in general. We don't know whether or not that is allowing the immune system to attack the cancer any better, but we do know that following those three elements, food, meals, sleep, and activity, exercise, are very important in general for outcomes for patients.
Perfect, thank you. The next question is, can you speak to anti-angiogenesis type drugs and if they are effective in preventing new capillaries from developing and attaching to cancer cells?
Yeah, so anti-angiogenic therapy. So angiogenesis is the process of forming new blood vessels, and this is a key important component for cancer. It needs to get its food, right? And it gets its food through the blood vessels. And there are drugs that can block the formation of those blood vessels. And the idea on one side is to help to prevent the cancer from getting the appropriate nutrients it needs. It also may actually change the biology of the cancer so that the immune system may be able to get in better into the cancer. Now, that all said, what does angiogenesis look like in pancreatic cancer? For the vast majority of pancreatic cancers, what we've learned is that angiogenesis is not quite as robust as it is for other cancers like kidney cancer and melanoma. It actually turns out that there are often very few blood vessels that are present within pancreatic cancer, and when they're present, they appear to be collapsed. And the the collapsing of those blood vessels appears to be because there's strong pressures inside the cancer that are pushing things out. It's helping the cancer to move out into the bloodstream. There have been several clinical trials testing anti-angiogenic therapies in combination with chemotherapy that were conducted many years ago. Unfortunately, those trials were not successful, but we've also more recently begun to learn that not all pancreatic cancers are the same, and some have more of this fibrosis and less blood vessels, and others have more blood vessels and less fibrosis. And so at this point, we don't know whether or not there is a role for anti-angiogenic therapies in pancreatic cancer, particularly for those patients that have more blood vessels and less fibrosis. These are areas of investigation in the laboratory, and they are areas of investigation in other cancers that behave similar to pancreatic cancer. And anti-angiogenic therapies are being combined with immune therapy to try to make it work better. So hopefully in the future we will learn more about how to use anti-angiogenic therapies and whether or not they will be a valuable treatment option for patients with pancreatic cancer.
Thank you for that explanation. The next question is, will patients experience more side effects if they're treated with immunotherapy and chemotherapy simultaneously? And in regards to immunotherapy, how do you keep patients healthy to receive that treatment?
Yeah, so this is a good question. The side effects can be a bit different. It can be challenging trying to understand what are the causes of side effects that occur in patients with pancreatic cancer in general. The abdominal pain and liver enzyme changes can occur due to the pancreatic cancer. They could occur due to the chemotherapy. They could also occur due to the immunotherapy. And so this becomes a dialogue with your treating physician to try to understand what is the cause of side effects that you're experiencing. Typically at this point, there are not prophylactic or preventative measures that are used beyond Tylenol and Benadryl and some other over-the-counter medicines to help during the infusion of the immunotherapy. So there's not preventative measures to try to prevent side effects that occur from the immunotherapy. In general, though, what we have seen is that by appropriate sequencing of the chemotherapy and the immune therapies, that many patients are able to tolerate the combinations well. But it becomes it's challenging, truthfully so, that understanding what is causing what, and we need to do better in terms of understanding how to prevent these treatments from causing side effects and whether or not that prevention will impair the ability of the treatment to work. And that remains a big question within the field.
Thank you for sharing that as well. The last question will be a two-part question. So how can patients find immunotherapy clinical trials? And in regards to today's time, is immunotherapy encouraged as a safe treatment option during COVID-19?
Yeah, so two very good questions here. So in terms of finding clinical trials, there are, for what I use to find clinical trials as a physician, but also can be used as a patient, are two resources. The first is that the Pancreatic Cancer Action Network offers a clinical trial resource for patients as well as physicians. And so you can log on and find clinical trial options there. Those are very much specific to pancreatic cancer. There's also the government through a website called clinicaltrials.gov also offers a listing of all clinical trials that are being conducted. And you can search for pancreatic cancer trials, you can search for immunotherapy trials, and you can use that as a resource to be able to discuss with your treating physician to identify the appropriate trial, that's a trial that's appropriate. The use of immunotherapy during this COVID-19 crisis. So I think that there's a lot that we know at this point, and there's a lot that we don't know. We know that COVID-19 is a virus. We know that it can activate our immune system. In many cases, some of the outcomes that are occurring very much well may be due to overactivation of the immune system. We know that some signs that occur in patients is that their T cell counts in the blood drop. They decrease, and that is consistent with what we will sometimes see with a viral infection. There have been precautions that have been looked at in terms of immunotherapy and in those patients who, in this setting of COVID-19. I would say at this point in time, there's no definitive evidence to suggest that receiving immunotherapy will cause a worse outcome. But I will also say that this is a topic that is very much at the forefront of physicians and thinking process, and that this becomes a personalized medicine strategy of identifying the risks and benefits of receiving therapy, and that's a dialogue that one needs to have happen with their treating physician. It's not going to be the same for everyone, and so there are precautions that are being put in place in terms of if one has been exposed recently, how should you wait in terms of the timing of your treatments. And those are individualized and personalized strategies that are being done at this point. There are no general guidelines at this time.
Thank you, Doctor. That was very helpful. At this time, we would like to thank Dr. Beatty for his very informative presentation. There may be a delay of several days before the information is posted, and your patience is appreciated. Once again, we would also like to thank our webinar sponsors, AstraZeneca and Ipsen, and our scientific and medical affairs industry members, AbbVie, AngioDynamics, AstraZeneca, Ipsen, Janssen Oncology, Pfizer, Rafael Pharmaceuticals, TriSalus, and TYME. If you have questions, please contact Patient Central at 877-2-PanCAN and ask to speak with an associate. This concludes the webinar. Thank you.